Welcome to another deep dive. Today, we're plunging into a scientific discovery that really dramatically reshaped our understanding of life itself. It's pretty remarkable how one breakthrough can just shift everything, isn't it?
It truly is, yeah. New knowledge has this incredible power, to reframe how we see the world. But, well, with that power comes responsibility, a big one.
Absolutely.
Oh.
Because powerful ideas can be, well, devastatingly misapplied.
hmm When you strip knowledge of the ethics of critical thinking, it just gets twisted. Used to push really harmful, prejudice-based agendas. We've seen it happen.
It's a stark reminder that just understanding something isn't enough. You have to get the context, the potential consequences.
Exactly. Which brings us right to our topic today. The history of genetics.
Yeah.
It's fascinating, but also parts of it are deeply, deeply unsettling.
Yeah, we're going to explore the maybe surprising misuse of Mendel's laws and how that kind of spiraled into the whole eugenics movement.
And then, after grappling with that really difficult history, it's even more critical to understand the actual science. So we'll get into the fundamental laws of population genetics like Hardy-Weinberg.
And the powerful forces that, really drive evolution.
Right.
So our mission for you listening in is to unpack how these simple genetic ideas got so twisted. Then explore how populations genuinely change over time.
And reflect on how our own human traits, our choices, fit into this bigger evolutionary picture.
Yeah, get ready to connect some surprising dots, maybe rethink some common assumptions.
Yeah.
Let's begin.
Let's dive in.
Okay, so to kick things off, let's rewind. Late 19th century, there's this key figure, Francis Galton. He was, interestingly, Charles Darwin's cousin.
Yeah, Galton. He coined the term eugenics back in 1883, and it literally means, good in birth or good stock.
And on the surface, the stated goal, it almost sounded, well, harmless, maybe even benevolent. The betterment of the human race.
Sure. It did seem that way to some. The idea was basically encourage people with what they decided were good characteristics, intelligence, certain looks to have more kids, aiming to, improve humanity.
But what was the huge flaw right from the start?
Oh, a massive one. It was just immense oversimplification. They assumed that really complex traits were purely hereditary, passed down, just like.
Like eye color or something.
Exactly, like eye color. There was zero rigorous science behind that assumption for things like intelligence or behavior.
And that's where Mendel comes in, right? His laws being rediscovered around 1900, that fueled this.
Precisely. It gave eugenics this false veneer of scientific backing. Mendel's simple rules for inheritance in pea plants, they were totally oversimplified and just wrongly applied to incredibly complex human stuff.
Like intelligence or musical talent?
Even job skills.
Yeah, all of it. They acted like these things were controlled by single genes following simple patterns.
And the evidence they used must have been shaky. Oh, completely. Mostly just anecdotal family trees, genealogies. They'd point to generations of shipbuilders or musicians in one family and say, see, it runs in the family. It's hereditary.
Not exactly rigorous science.
Not even close.
Yeah.
But it gave these dangerous ideas a powerful credibility they didn't deserve.
And this thinking, it clearly took root fast in America.
It really did. By 1906, you had the American Breeder Association forming a specific eugenics organization. They were actually using Punnett squares.
With Punnett squares, like we learned in high school biology.
Yep. To demonstrate the probabilities of having, quote, good versus less intelligent children. It's chilling.
And they explicitly tied this to race, didn't they?
Oh, absolutely. With
Yeah.
just appalling pseudoscience. Claims like the gene for white intelligence was dominant and black intelligence was recessive, warning that race mixing was a disservice to humanity. It's just awful.
Wow. And this sounds like it fed directly into things like those fitter family contests you hear about from the 1920s.
Directly. Families judged on looks, physical appearance, mental capacity, and the really unsettling part. Only white families ever won. It just reinforced all the existing prejudices. That was their idea of positive eugenics, get the fit to reproduce more.
But then there was the darker side, negative eugenics.
Right. This was about preventing reproduction among those they deemed unfit or having less desirable traits. The grim logic was that these people would, drag down the quality of the human race.
And this wasn't just talk, it became actual policy, horrifyingly.
Indeed. Indiana passed the very first compulsory sterilization law in the U.S. way back in 1907. By the 1930s, about 30 U.S. states had similar laws.
Targeting who exactly?
People labeled imbeciles,
Yeah.
feeble-minded, certain criminals, and the criteria could be unbelievably flimsy. Failing 10th grade 3 times in some places, that could be grounds for forced sterilization.
That's unbelievable. And the Supreme Court actually upheld this in Buck v. Bill.
Yeah, 1927. Infamous case. Carrie Buck, her mother, her baby daughter, all declared feeble-minded based on really weak, biased tests. Justice Oliver Wendell Holmes wrote the opinion.
And that chilling line.
3 generations of imbeciles are enough. It just perfectly captures that cold, deterministic mindset of the era, believing these things were just locked in by genes.
And these laws weren't just a blip. They stayed on the books in some places until the 1980s.
That's right, resulting in over 60,000 people forcibly sterilized in the U.S. alone. And the definition of undesirable just kept expanding. Alcoholism, criminality, even poverty, all framed as primarily genetic issues.
It even bled into immigration policy, didn't it? The Johnson-Reed Act in 1924.
Yes, explicitly designed to limit immigration from Eastern and Southern Europe, non-Nordic people, based on completely unfounded claims about them having bad genetic qualities. It was racism, pure and simple, just dressed up as science.
And then, of course, the most horrific manifestation, Nazi Germany.
Unquestionably, the Holocaust. Systematic persecution, murder, targeting Jewish people, people with disabilities, Roma people, political opponents, all based on this hateful, arbitrary ideology. Even things like being short or having moles could mark you for death.
You even had programs like Lebensborn trying to actively breed certain traits.
Exactly, a selective breeding program encouraging so-called pure Aryan women to have children with SS soldiers, literally trying to breed loyal Germans. They genuinely believed traits like nationalism were genetic.
It's just grotesque. And the scientific experiments.
Some were truly horrific, like Eugene Fisher's work in Namibia, forcing German men to have children with local African women to study racial hybrids, and then executing the children to collect their skulls for study. Science utterly perverted.
Thankfully, after World War II, things started to shift.
Yes, the sheer scale of the Holocaust in humanity became undeniable, especially post-1945. It forced a global reckoning with eugenics.
And at the same time, genetics itself was advancing, moving beyond those simplistic ideas. We started learning about polygenic inheritance.
Correct. The discovery that most complex traits, intelligence, height, things like that, aren't controlled by single genes. They're influenced by many interacting genes and, crucially, the environment.
The environment factor. So it's not just the genes.
Not at all. Think of it like this. Your genes might be the hardware, say, a really powerful computer, like a Tesla car maybe, but the environment, that's the fuel, the road conditions, the software updates. Bad roads, bad fuel, even a Tesla won't perform well.
Or maybe a different analogy, like a sturdy SUV, a Fortuner, might actually do better on those bad roads than the fancy sports car.
That's a great way to put it. The hardware matters, but its performance totally depends on the environment it's operating in. It's not rigid biological determinism, genes dictating everything.
It's a complex interplay. Genes and environment.
Exactly. And understanding that shift was and is so important. It's a devastating historical lesson, really, on why we need proper scientific understanding and serious ethical thought.
Which is why, to truly avoid repeating those kinds of mistakes, we need to grasp the actual science of how populations change genetically. So let's shift gears now. Let's talk population genetics, the real building blocks of evolution.
Okay, population genetics. So instead of just tracking traits from parent to kid, this is looking at the bigger picture, right? The whole group.
Exactly. It applies those basic genetic principles, but to understand the entire gene pool of a population, not just single individuals. Because that's the level where evolution actually happens in populations over time.
And to study this, scientists use this idea of a Mendelian population. What exactly is that? Is it like a specific town?
It's more of a theoretical tool, but yeah, it can be defined geographically. It basically means a group of individuals who are much more likely to mate within that group than outside it. It sets up a kind of boundary for gene flow.
Like a sort of closed-off genetic circle.
Pretty much. So a whole species could be one huge Mendelian population. But you also have subgroups, like people in India might primarily mate with others in India versus people in Papua New Guinea. Geography creates a boundary.
Okay, that makes sense. But it's not just geography, right?
Culture can create these boundaries too. Nationality, religion, caste systems, even income levels can lead to groups primarily mating within themselves. They effectively become distinct Mendelian populations, each with its own gene pool.
Gotcha. And the gene pool itself is just all the genes.
All the alleles, yeah.
Yeah.
All the different versions of every gene carried by every single member of that defined population, just the sum total.
Okay. So how do we measure what's in the gene pool? Frequencies.
Exactly. Two main types. First, genotype frequency. That's just the proportion of individuals who have a specific genetic makeup. Like if 16 out of 100 people are genotype AA, the frequency of AA is 0.16 or 16%. You're just counting heads with specific genotypes.
Simple enough. And the other one, allele frequency or gene frequency. This tells you how common a specific allele, like the dominant A allele or the recessive A allele, is within that entire gene pool. You basically count up all the copies of that specific allele across everyone and divide by the total number of copies of that gene in the population. It's just math, really.
Okay. And this leads us to how we define evolution in these terms. It's not just, apes turning into humans over millennia.
In genetic terms, it's much more precise. Evolution is simply the change in gene or allele frequencies within a population over time, from one generation to the next.
So if those frequencies, the percentage of alleles and alleles, stay exactly the same generation after generation,
Then the population is in genetic equilibrium. It's stable. It's not evolving, at least for that particular gene. And this brings us to a really important concept, the Hardy-Weinberg Equilibrium.
right, named after Godfrey Hardy, a British mathematician, and Wilhelm Weinberg, a German physician. They figured this out independently. What's the core idea?
The core principle is basically describing that state of non-evolution. It says that gene and genotype frequencies will remain constant generation after generation if, and this is a big if certain conditions are met and if no evolutionary forces are messing things up.
So it's like a theoretical baseline, a benchmark.
Exactly. It tells us what things would look like if evolution wasn't happening. So if we look at a real population and find its frequencies don't match the Hardy-Weinberg predictions,
Then we know evolution is happening. Something is causing those frequencies to change.
okay, so what are these conditions needed for perfect equilibrium, the no evolution state?
Right, there are a few key ones, and you'll see they're pretty unlikely in the real world. First, the population has to be really large. This is to avoid random chance fluctuations in frequencies, which we call genetic drift.
Okay, large population. What else?
You need roughly equal numbers of males and females. And third, everyone has to be equally fertile and have an equal chance of reproducing. No differential success. Everybody contributes equally to the next generation's gene pool. That already sounds unrealistic. Equal reproductive success.
Highly unrealistic. And the fourth condition is totally random mating. Any individual has an equal chance of mating with any other individual in the population, regardless of their genotype or appearance.
Yeah, okay. Large size, equal sexes, equal fertility, random mating. That basically never happens perfectly in nature, does it?
Almost never, which is why evolution is pretty much the constant state for most populations. Now, mathematically, Hardy-Weinberg gives us a simple way to relate allele frequencies to genotype frequencies under these ideal conditions.
The P's and Q's.
Right. If P is the frequency of the dominant allele, say A, and Q is the frequency of the recessive allele, then P plus Q must equal 1. They have to add up to 100%.
Makes sense. And for the genotypes?
Under random mating, the expected genotype frequencies are P squared for the homozygous dominant, AA, 2PQ for the heterozygous AA, and Q squared for the homozygous recessive A. And again, P2 plus 2PQ plus Q2 must equal 1.
So let's say P is 0.5 and Q is 0.5. We'd expect frequencies of 0.25 LA, 2.50 LA, and 0.25 AA.
Exactly. Now, if you go out and measure a real population and find, say, 0.40A, airborne 0.40A, 0.40, and 0.40 AA.
Then something's up. The frequencies don't match the prediction.
Right. It tells you that one or more of those Hardy-Weinberg conditions isn't being met. An evolutionary force is at work, changing the frequencies.
Okay, so that's the crucial next step. What are those forces? What disrupts that perfect Hardy-Weinberg balance and actually drives evolutionary change?
Yeah, these are generally considered the 5 major forces or agents of evolution. They're the things that cause gene and genotype frequencies to change over time, pushing populations out of that theoretical equilibrium.
Let's break them down. Force number 1, mutation.
Right. Mutation is the ultimate source of all new genetic variation. It's any random change in the DNA sequence.
So this is where new alleles come from in the first place.
Exactly. It's the only way new alleles are created. All the other forces, selection, drift, etc., they act on the variation that mutation provides. Think about the sickle cell allele we mentioned earlier. That arose from a single DNA change, a mutation.
And it causes disease if you have 2 copies. but offers malaria resistance if you have one. Shows that the context matters.
Hugely. Okay, force number 2. Natural selection, Darwin's big idea, often called survival of the fittest.
But it's more nuanced than that, right?
Much more. It's really about differential reproductive success. Individuals who happen to have variations that arose from mutation that make them better suited to their current environment are more likely to survive and reproduce.
So they pass on those advantageous variations more often. Nature selects what works best right here, right now.
Precisely. And selection can operate in different ways. For instance, there's stabilizing selection.
What's that? Favoring the middle.
Exactly. It favors the average or median phenotype and selects against the extremes. Human birth weight is the classic example. Babies born too small or too large historically had lower survival rates. So selection favors weights closer to the average. It keeps the population centered, sometimes called balanced polymorphism.
Okay. Then there's directional selection.
This happens when the environment changes, or maybe a new mutation provides a significant advantage. Selection then favors one extreme phenotype, shifting the population's average trait value in that direction.
Like the peppered moths in England during the Industrial Revolution, the darker ones became more common.
Perfect example. Pollution darkened the trees, so darker moths had better camouflage from birds. Other examples. Antibiotic resistance in bacteria, strong directional selection there.
Right, because the antibiotics kill off the non-resistant one.
Exactly. Also, the steady increase in human brain size over our evolutionary history, or the evolution of darker skin pigmentation in populations living in high UV environments, or lactase persistence, the ability to digest milk sugar as an adult in populations with a history of dairy farming. All directional selection.
And the third type, disruptive selection.
This one's interesting. It favors both extremes of a trait and selects against the average or intermediate forms. It can potentially even split a population into two distinct groups over time.
Any examples?
A hypothetical one often uses squirrels, where, say, short tails are good for maneuvering on the ground, long tails are good for balance in trees, but medium tails offer no particular advantage in either environment. Selection would favor the short-tailed and the long-tailed squirrels.
Got it. What about something like that CCR5 Delta 32 variant? The one linked to HIV resistance. How does selection work there?
That's a really complex and fascinating case. So this variant is a deletion in the CCR5 gene. It seems likely it was originally selected for because it offered resistance to other diseases, maybe smallpox or plague, centuries ago in Europe.
Okay, so it had a past advantage.
Possibly. Now, in the context of HIV, having one copy of the deletion, being heterozygous, gives some partial resistance to HIV infection. Having two copies, homozygous, provides almost complete resistance.
So selection would favor it now in areas with HIV.
You'd think so, and it might be slowly increasing in some populations. But it's complex. It also highlights how past selective pressures matter. For instance, this variant is very rare in places like India, which unfortunately correlates with higher HIV susceptibility in those populations today. It shows how history shapes vulnerability.
Wow. Okay, force number 3, sexual selection. How is this different from natural selection?
It's really a special case of natural selection. Here, traits are selected for specifically because they increase an individual's success in, well, finding mates and reproducing. It's not necessarily about survival directly, but about mating success.
Like the classic example, the peacock's tail.
Exactly. That massive, colorful tail is metabolically expensive to grow, makes the male more visible to predators. It doesn't seem great for survival, but peons find it incredibly attractive.
So males with the biggest, most elaborate tails get chosen more often.
And reproduce more, passing on the genes for those elaborate tails. The reproductive payoff outweighs the survival cost. That's intersexual selection or female choice.
And there's another type, intrasexual selection.
Right. This is usually competition within one sex, typically males for access to mates, usually females. Traits that help males win these competitions get favored.
Like male deer locking antlers.
Perfect example. Or physical strength and physique in many species, including, historically, humans. Stronger males might have been perceived as better protectors or providers, giving them an edge in competing for mates.
This sounds like it gets into evolutionary psychology territory, looking at our mating preferences today.
It definitely does. David Buss did famous cross-cultural studies looking at what men and women value in long-term partners across dozens of different societies.
And what did he find? Were there consistent patterns?
Surprisingly consistent, yeah. Across cultures, men tended to place a higher value on youth and physical attractiveness in long-term partners, interpreted as cues for health and fertility or reproductive potential.
Okay. And women?
Women, again across diverse cultures, tended to prioritize financial prospects, education, ambition, emotional stability, and even humor in long-term partners. Things that might indicate resources, protection, and commitment for raising offspring.
Interesting. But how much does that deep evolutionary history still shape us today? Society's changed so much. Women's financial independence, for example.
That's the million-dollar question, isn't it? It's a huge area of debate and ongoing research. Preferences are evolving, they're complex, culturally influenced, but those evolutionary echoes are likely still there, interacting with modern life. It's something really fascinating for you, the listener, to think about in your own observations.
Definitely food for thought. Okay, let's move to the fourth force. Genetic drift. This one's about randomness, right? Not selection.
Exactly. Genetic drift is all about random fluctuations in allele frequencies from one generation to the next, happening purely by chance. It's not because an allele is better or worse, just luck of the draw.
And it's especially powerful in small populations.
That's the key factor. In a small population, random events can have a disproportionately large impact on which alleles happen to get passed on. Imagine flipping a coin only 10 times versus 1,000 times. You're much more likely to get a skewed result with only 10 flips.
Makes sense. Are there specific scenarios where drift is really obvious?
Two classic scenarios. First is the founder effect. This happens when a small number of individuals break off from a larger population and go start a new isolated population somewhere else.
So the founders might just randomly have different allele frequencies than the original big group.
Precisely. By pure chance, their small gene pool might be unrepresentative. A famous example is the relatively high frequency of the O blood group among many Native American populations compared to the East Asian populations they originated from. It likely reflects the specific blood types of the small founding groups that crossed the Bering Land Bridge.
Or that island, Tristan da Cunha.
Another great example. A very rare dominant allele causing a form of blindness, retinitis pigmentosa, is at a surprisingly high frequency there, traced back to one of the original 15 founders in the early 1800s. Pure chance in the founding event.
Okay, founder effect. What's the second scenario? Bottleneck effect.
The bottleneck effect. This happens when a large population suffers a drastic reduction in size, maybe due to a natural disaster, disease outbreak, overhunting, just a catastrophic event that randomly kills off most individuals.
So the few survivors become the bottleneck.
Exactly. And the gene pool of those few survivors might, again, just by chance, be very different in its allele frequencies from the original large population. They're the lucky ones who made it through, not necessarily the fittest.
Any examples of bottlenecks?
The Pinjalap Atoll in Micronesia is a striking one. A typhoon devastated the island in the 1770s, leaving only about 20 survivors. One of those survivors happened to carry a rare recessive allele for complete color blindness, or chromatopsia. Now, due to drift following that bottleneck and subsequent inbreeding, a significant percentage of the Pinjalupese population has this condition. It's far, far higher than anywhere else in the world. It shows how random events can dramatically reshape a gene pool.
So the takeaway from drift is, it's not always survival of the fittest. Sometimes it's just survival of the luckiest.
That sums it up perfectly. Chance plays a big role, especially in small populations.
Okay, on to our fifth and final force, inbreeding.
Right. So inbreeding directly violates that Hardy-Weinberg condition of random mating.
How is it defined?
It's non-random mating between individuals who are more closely related than you'd expect just by chance in the population, or sometimes it refers to mating between individuals with similar genetic features, which also increases homozygosity.
And the main negative effect, short-term at least, is that increase in homozygosity, having two identical copies of an allele.
Exactly. It brings together two copies of the same allele inherited from that common ancestor. Now, if those alleles happen to be recessive and deleterious, meaning they cause genetic disorders when you have two copies inbreeding, dramatically increases the chances of those disorders showing up.
Like we see in, say, pedigree, dogs bred for specific traits, or historically in isolated royal families.
Precisely. This leads to what's called inbreeding depression, a general reduction in the population's fitness, survival rates, fertility, and overall adaptability.
Are there any upsides? from an evolutionary perspective?
Well, from a very long-term theoretical view, by increasing homozygosity, inbreeding can bring those harmful recessive alleles out into the open, making them visible to natural selection, which can then potentially eliminate them, sometimes called a purging effect. It can also increase homozygosity of potentially advantageous alleles.
But the main outcome, especially in the shorter term.
The main outcome is usually a reduction in overall genetic diversity, and diversity is generally crucial for a population's ability to adapt to future environmental changes or new diseases.
So diversity is strength, evolutionarily speaking.
Absolutely. Losing diversity through things like drift or inbreeding often makes a population more vulnerable.
Wow. Okay, we have covered a lot of ground. We started with the really shocking misuse of basic genetics in the eugenics movement.
Yeah, a truly devastating historical lesson, how flawed interpretations, stripped of ethics, fueled by prejudice, led to horrifying policies, compulsory sterilization, racist immigration laws, and ultimately contributing to the ideology behind the Holocaust.
Then we shifted gears, understanding the actual science of population genetics, the gene pool, allele frequencies.
And Hardy-Weinberg Equilibrium as that crucial baseline, that state of no evolution against which we measure change.
And finally, the 5 key forces that do cause change, that sculpt gene pools constantly. Mutation bringing in the new variations.
Natural and sexual selection favoring traits that work for survival or reproduction in a given context.
Genetic drift introducing random chance fluctuations, especially in small populations.
And inbreeding, the non-random mating that reduces diversity. It's this complex interplay always happening.
It really drives home that evolution isn't just some abstract thing that happened millions of years ago.
Not at all. It's a dynamic, ongoing process. It's happening right now in all living populations, including us humans. Our gene pools are subtly shifting constantly under these pressures.
And that brings us back to that initial point about responsibility. Scientific knowledge is incredibly powerful.
Immensely powerful. But like any tool that powerful, it demands immense care, ethical reflection, critical thinking. We have to ensure it's used to benefit humanity, not harm it. Eugenics stands as the ultimate, terrible, cautionary tale.
So maybe a final thought for you, the listener, to ponder. As you go about your life, observing human behavior, societal trends, health patterns, even reflecting on your own choices and preferences.
Ask yourself, how might these echoes of evolution, these subtle ongoing shifts in our shared human gene pool, driven by the very forces we've just discussed, be playing a quiet role in the world you see around you today?